Biology with Dr. E.
We now turn our attention to proteins. Proteins comprise approximately 10 to 30% of the cell's mass. They contain carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur and phosphorus. Proteins have the most varied functions of any molecule. We will review the functions of proteins on the next slides. Let's review some functions of proteins. Structural proteins provide mechanical support. For example, collagen, found in all connective tissue, is the most abundant protein in the body. It is responsible for the tensile strength of bones, tendons, and ligaments. Enzymes are biological catalysts. Enzymes are essential for virtually every biochemical reaction in the body. For example, proteases hydrolyze proteins. Here you see an enzyme catalyzing the breakdown of a disaccharide into individual monosaccharides. Transport proteins help move substances in blood and across the membranes. For example, hemoglobin transports oxygen in blood. Some plasma membrane proteins transport substances such as ions across the plasma membrane. Contractile proteins enhance movement. For example, actin and myosin cause muscle cell contraction and function in cell division in all cell types. Communication proteins function in transmitting signals between cells. They can act as chemical messengers, for example, hormones, or as receptors in the plasma membrane. Defensive proteins protect against disease. For example, antibodies released by certain immune cells are specialized proteins that bind and inactivate foreign substances such as bacteria, toxins, and viruses. Proteins are polymers of amino acid monomers held together by a special covalent bond called the peptide bond. So because of this, proteins are generally called peptides. Polypeptides are 10 or more amino acid chains bonded together. Proteins are up to 50 or more amino acid chains bonded together and each held by peptide bonds. There are 20 different types of amino acids, and each amino acid has an amino group and a carboxyl group. Because of these two functional groups, an amino acid can act as either an acid or a base. Each amino acid has a side group represented here by R. This side group determines the specific properties of the amino acid. This figure illustrates formation of a dipeptide. In dehydration synthesis, the acid group of one amino acid is bonded to the amine group of the next amino acid with loss of one water molecule. This forms a dipeptide, which consists of two amino acids held together by a peptide bond. The dipeptide can be broken down into two of the component amino acids by hydrolysis. In hydrolysis, the peptide bond that links amino acids together are broken down when a water molecule is added to the bond. The dipeptide breaks apart into two amino acids. Next, let's review the structural levels of proteins. The shape and function of proteins is determined by four structural levels: primary, secondary, tertiary, and quaternary. We'll review each of them over the next couple of slides. This figure illustrates the primary structure of a protein. In the primary structure of a protein, the sequence of amino acids forms a linear polypeptide chain that serves as the backbone of the protein structure. This figure illustrates the secondary structure of a protein, which forms when the primary chain twists into spirals or sheets. When the primary structure twists into spirals, the resulting coil structure is called an alpha helix. Alpha helices are stabilized by hydrogen bonds, shown here as dots. When the primary structure bends into sheets, it zigzags back and forth to form a pleated structure called a beta sheet. Adjacent strands of beta pleated sheets are held by hydrogen bonds, shown here as dotted lines. This figure illustrates the tertiary structure of a protein, which is superimposed on its secondary structure. The alpha helices and beta pleated sheets are folded up to form a compact globular molecule held together by intramolecular bonds. The figure illustrates the tertiary structure of transthyretin, a protein that transports the thyroid hormone thyroxine in blood and cerebrospinal fluid. Here we see the quaternary structure of a protein. In the quaternary structure, two or more polypeptide chains, each with its own tertiary structure, combine to form a functional protein. The figure constitutes the example of transthyretin by showing how four identical transthyretin subunits join to form a complex protein. Proteins can be classified as fibrous or globular proteins. Fibrous proteins are also called structural proteins. They are typically strand-like, water-insoluble, and very stable. Most have tertiary and quaternary structure, and their main function is to provide mechanical support and tensile strength. Examples are keratin, elastin, collagen, and some contractile proteins. Globular proteins are also called functional proteins. They are compact, spherical, and water-soluble, and very sensitive to environmental changes such as pH and temperature. They also have tertiary or quaternary structures. Most globular proteins have specific functional regions called active sites. Examples include antibodies, hormones, enzymes, and more. What happens when the structure of a protein is altered? Please note that each amino acid has a unique three-dimensional shape. Substitution of one or more amino acids may change the shape and function of a protein. For example, in sickle cell disease, substitution of valine for glutamic acid on one chain of the hemoglobin molecule causes abnormalities in the red blood cell function. Protein denaturation. Protein denaturation is a process in which a protein loses its three-dimensional structure, resulting in loss of function. Globular proteins unfold and lose their functional shape. Fibrous proteins are more stable. During denaturation, the active site becomes deactivated. Denaturation could be caused by changes in pH and temperature. In some cases, denaturation could be reversible if normal conditions are restored. However, in extreme circumstances, this process is irreversible. For example, if you had boil an egg, there is no way you can reverse that process. Enzymes and enzyme activity. Enzymes are globular proteins that act as biological catalysts. Catalysts regulate and increase the speed of chemical reactions without being used in the process. Enzymes lower the activation energy needed to initiate chemical reactions. This leads to an increase in the speed of reaction and allows for millions of reactions per minute. Characteristics of enzymes. Most functional enzymes are referred to as holoenzymes. consisting of two parts, an apoenzyme, or the protein portion of the enzyme, and either a cofactor or a coenzyme. Cofactors are usually metal ions. Coenzymes are often organic molecules, usually vitamins. Enzymes are very specific in their action. That's because they can only act on very specific substrates. The names of enzymes usually ends with "ase", indicating the type of reaction they catalyze. For example, hydrolase and oxidase. Enzymes lower activation energy, which is the energy needed to initiate a chemical reaction. Enzymes allow chemical reactions to proceed quickly at body temperatures. This figure consists of two charts. Both charts show reactants entering a reaction, with a curve indicating the amount of activation energy required for the reaction to occur and for products to be formed. The first chart, indicated here, shows the reaction without an enzyme being present. You will see there's a steep curve showing that a great deal of activation energy is necessary for the reaction to occur and continue. The second chart here shows the reaction when the enzyme is present. The curve is much lower, indicating that much less activation energy is required to prime the reaction. How do enzymes bring about their action? This figure illustrates the three steps involved in enzyme action. In the first step, the substrate, in this case amino acids, bind to the active site of the enzyme, temporarily forming an enzyme-substrate complex. In the second step, the enzyme-substrate complex undergoes internal rearrangement that forms the product. A bond is formed between the amino acid, absorbing stored energy and releasing water in the process. In the third stage, the enzyme releases the product of the reaction. The product is a dipeptide formed by the formation of a peptide bond. The reaction also releases the enzyme, which returns to its original shape and is available to catalyze another reaction.